Greetings everyone. So today we are going to discuss about a new topic which is the structure of HIV. Before talking about HIV, let us discuss about what is HIV, its introduction. So HIV, previously told, it is a retrovirus. It belongs to the lentivirus subgroup of the retroviruses. Retroviruses are those viruses which have a reverse transcriptase enzyme and perform reverse transcription. These viruses usually cause tumors, so these are also known as oncoviruses, as they are having their tumors, so these are known as oncoviruses, as well as these viruses have the characteristic of causing tumor, as well as these are mostly RNA viruses. When it comes to the structure of HIV, particularly HIV is spherical in shape. It is spherical in shape, as well as it is a virus which is enveloped virus, as well as this kind of virus is enveloped virus, and it contains three types of enzymes known as integrase, reverse transcriptase. In short, we can write it as RTase, or in long, we can write it as RNA dependent DNA polymerase. It is a multifunctional enzyme, RTase, because it performs two to three functions. It is in case a regulatory enzyme because it performs two to three functions over here. So it is a regulatory enzyme. On the other hand side, uh, it also possesses some another enzyme which is known as protease. Uh, Here, as you can see in the diagram, we are going to explain the diagram as well, that how are these numbers P17, P24 labeled. Moreover, coming to its general structure is derived into four parts. First part includes the envelope. Envelope is a part which includes the lipid bilayer. There's two lipids of lipid. There are two layers of lipid, which is known as lipid bilayers. These lipid bilayer are present, and due to the presence of lipid bilayer, they are more sensitive to temperature, as discussed in our video about why viruses survive in the host cell and how do they do so. So we are going to have that video as well. But for now, here we are going to focus on this thing, the lipid bilayer. Here they are having lipid bilayer. Within the lipid bilayer, there are several spikes arising. Several spikes are arising, which are known as the glycoprotein spikes, GP spikes. Specifically, when we come to their structure of this glycoprotein spike, it is divided in such a manner that this downward part, this transmembrane glycoprotein, which is embedded within the lipid bilayer completely, is known as GP41. GP41, glycoprotein 41, because it is having molecular weight as 41 kilodalton, and that is basically um, uh, we call this as transmembrane protein, as well as it helps in the fusion of the lipid bilayer of HIV with lipid bilayer of the host cell. When it comes to the other part over here, which is the outer protruded part, uh, which is known as GP120. Again, 120 for its molecular weight, 120 kilodalton, and here GP120 represents the outer protruded part. It is known as the head receptor. It acts as a receptor. It helps in attachment, as well as it also helps in attachment or binding, and acts as a receptor. Basically, it is a docking protein because it is protruded out from its side. Moreover, there are some other structures present, such as number one included envelopes. Number two here includes matrix. Matrix is a part, it is a proteinaceous matrix which basically provides stability to the capsid which is present over here. So this matrix is basically made up of a special type of protein which is known as P24 protein. P24, if I'm not wrong. This is made up of it's made up of P17 protein, while the capsid is made up of P24 protein. Once again, it's a mistake. So here it is made up of P17 protein, known as capsid, and down towards the capsid basically is stabilized by the matrix, and matrix is made up of P17 protein, whereas capsid is made up of P24 protein. These are the capsomer units which of which it is made up of. Moreover, when it comes to uh, the inner part after the capsid, we are having one more thing, that matrix plus capsid forms the protein shell. We can collectively call them as protein shell. Apart from which, the fourth part over here involves the genome, the core. The core basically involves nucleotides as well as some proteinaceous enzymes. Proteinaceous enzymes are there. Basically, first let us discuss about the genome. Genome basically consists of two identical single-stranded RNA, which are positive sense RNA 5 prime to 3 prime. These are identical, not complementary to each other, not antiparallel to each other. So these are the viral genome. Since it possesses two identical single-stranded RNA, it is known as a diploid virus because it is having two identical genome. So it is a diploid virus. Moreover, its structure is in discoided and folded form. Apart from which, when it comes to its enzymes here which are present are already told, which is integrase, protease, and RTase. When it comes to protease has its own function, and other enzymes also have their own function. Now, how does it work? We are going to discuss in our next slide. As you can see, there's a molecular weight of these enzymes as well. For example, if we are having protease, it is P10, which means 10 kilodalton is a molecular weight of protease enzyme. Reverse transcriptase is having its molecular weight as 64 kilodalton, whereas on the other hand side, we are having another structure over here, integrase, which is having its molecular weight as 32 kilodalton. So these are some things I wanted to discuss over here. Rest, when it comes to its life cycle, we are just going to discuss the intro of their life cycle. First includes the fusion. There are 7 steps of its life cycle, out of which the first step is fusion. Second step includes the first step is sorry binding or attachment. Second step is fusion. Third step is reverse transcription. Fourth step is integration. Fifth step is replication. Sixth step is assembly, and seventh step is budding. So this is a long process which we are going to discuss step by step in our next video, but now we are going to discuss an intro of it. We know that in our body there are cells, immune cells known as T lymphocytes. These T lymphocytes are matured in thymus, and these are developed in the bone marrow. These basically are differentiated into four types of cells in thymus, which includes T helper cells, T killer cells or cytotoxic T cells, T suppressor cells, and T memory cells. When it comes to T helper cells, macrophages, and dendritic cells present within our body are known as CD4 cells because they contain CD4 receptors. As they contain CD4 receptors, they also must have some co-receptors of CD4. For example, in macrophages are having co-receptor CCR5, whereas T helper cells are having CD4 co-receptor, which is known as C into CR4, C into CR4. So these are the core receptors present over here, which help the virus in fusion when the HIV virus is fusing its lipid bilayer with the lipid bilayer of the host cell, as a result of which fusion is possible and the virus is then able to take its protein shell and genome within the cell. Now, of course, there's a long process. We are going to discuss step by step. First, let us take an example of, for suppose if we are having T helper cell. T helper cell is having um, CD4 receptor, and here this CD4 receptor, as mentioned in the diagram, it is being attached with the GP120. GP120 and CD4 receptor attach in such a manner as if there is a lock and key model. Now, for suppose this is the CD4, this is basically the GP120 protrusion, while this part which I'm drawing right now showcases the CD4 receptor. So when they attach together in the form of a lock and key model, this lock and key model now basically this is kind of lock and key model, and that lock and key model, let me just draw it. So this is a lock and key model, and that lock and key model when attaches, it causes the this lock, which causes this lock causes this lock. All right, it causes this complete lock to move beside, to move turn aside. As a result, as it turns aside, it causes the lipid bilayer of HIV and lipid bilayer of host cell to combine and attach, or as a result of attaching, the co-receptor C into CR5 CR4 or CCR5 in case of macrophages are attached, are helping in the process of fusion by in such a manner that whenever the complex of the attached CD4 receptor at GP120 head moves aside, it causes the co-receptor CCR4 C into CR4 and CCR5 to vibrate the lipid bilayer. As a result of vibration of lipid bilayer, it causes the lipid bilayer to engulf the proteinaceous structure. It causes the lipid bilayer to fuse with the lipid bilayer of HIV, and thus it leads the proteinaceous structure of HIV to come within the host cell, due to which when it goes inside the host cell, the structure which is formed during fusion is just like this, as you can see in the diagram. This is the second step in which fusion occurs, and here HIV RNA reverse transcriptase integrates and other viral proteins enter the host cell. As it enters the host cell, the enzyme present here known as protease is utilized. Protease enzyme has dual role. First role is in the degradation of its own proteinaceous material, and second role is in the cleavage of large proteinaceous subunits which it makes and convert it into smaller proteinaceous subunits. For example, why is that so? This is because the proteinaceous subunits formed by the HIV are encoded by genes which code large proteinaceous subunits, due to which, uh, in order to cleave them, there is a need of an enzyme which is known as protease. So here, protease enzyme works, and protease enzyme causes to degrade the protein shell, the matrix, and capsid, as a result of which the genome as well as the enzymes are then entered the viral within the host cell, and thus the host cell is infected. Now when it is infected further, what does the virus do is that the viral RNA, which is single stranded RNA, of course, which is 5 prime to 3 prime. Now what it does is utilize the reverse transcriptase enzyme, as a result of which it forms a complementary strand. It forms a complementary DNA strand. It forms a complementary DNA strand which runs from 3 prime to 5 prime. This is RNA. Let me change the color as well. This is RNA strand. This is RNA. While on the other hand side, the blue one represents DNA. This is RNA strand. This is DNA strand. DNA strand. This is complementary DNA strand. Now at that moment, DNA and RNA strands are formed, and once they are formed using the RTase enzyme, there is also formation of hydrogen bonding between them. There's also formation of hydrogen bonding between them. So they form a complex in which RNA is also present, DNA is also present, which means a molecule which is having RNA and DNA. Now, in order to break the hydrogen bonding, the RTase enzyme also works as an RNase enzyme in order to cut the RNA and DNA strand by cleavaging its by cleaving its hydrogen bonding. As it cleaves the hydrogen bonding, as the RNA strand and DNA strand are broken by cleavage of the hydrogen bonding, it causes the hydrogen bonding to be broken, and thus both strands are separated, as a result of which the cDNA right now, the cDNA which is formed here, will now further utilized by RTase. Now this cDNA will further form its complementary DNA. cDNA now here is 3 prime to 5 prime. It will form its complementary DNA, which is 5 prime to 3 prime, which is 5 prime to 3 prime, and and that would be formed as a complementary strand. And again, the same enzyme RTase will be utilized, but this time RTase will also function as DNA dependent DNA polymerase rather than RNA dependent DNA polymerase. So we can say that complementary DNA strand of the cDNA formed by RNA is identical to the RNA strand, while it is complementary to the cDNA, or the complementary DNA formed by the RNA. So this is what we are going to discuss. Once the double-stranded DNA is formed, here integration occurs. Basically, the whole process which is explained over here will occur in the cytoplasm. Once the double-stranded DNA is formed from mRNA, sorry, not from mRNA. Once the RNA of viral genome of the HIV is converted in double-stranded DNA using the RTase enzyme. It is then sent within the nucleus from the cytoplasm, where the integration of the viral double-stranded DNA with the double-stranded DNA of the T helper cell occurs, as a result of which to integration leads uh, to incorporate the viral genome with the T helper cell genome. Now, enzyme utilized here is integrase. Now, once integration occurs, then the next step includes replication such that it produces genes of its desired protein. Now, of course, here, as you can see, that as it will start to replicate itself within the cell, it will not replicate as cleverly, because first, one more thing here, when the integrated DNA which is formed by the integration of viral genome and the T helper cell genome, it will be known as provirus. Now that provirus will tend to replicate, and once it will tend to replicate, this replication of provirus will lead to several copies of HIV, which would be sent in other cells. Initially, when a person is infected by HIV, the person doesn't show any symptom for many years, around 8 to 10 years, and after 8 to 10 years, it simultaneously shows various symptoms within the person, as a result of which it is a virus, which basically at first remains silent for many years, and after it is done with its work of after it knows that uh, after 8 to 10 years, it's then able to replicate itself, and once it is able to replicate itself, it then spreads all over the body, due to which it's difficult to treat it. One more thing over here, that whenever the HIV virus is replicated, HIV virus does not has a proofreading system, which means whenever we are replicating the eukaryotic DNA, the eukaryotic DNA once replicated is proofreaded, so that there are no chances of mutation left in the DNA. But when HIV replicates its DNA, it does not have such a system, due to which it leads to mutations always. It always makes a mistake, due to which we can say that every antigen has a different gene, which means there's mutation, antigenic variation is present. As a result of this antigenic variation, there are no vaccines. uh, there can be vaccines and somehow, but usually there are no vaccines available for HIV. So this is one more thing. And c into CR4 receptor is a kind of receptor which can be inhibited. What does that mean? Wait a minute. C into CR4 is a receptor which is found on T helper cell. So T helper cell contains a C into CR4 receptor, and CCR5 receptor is possessed by macrophages. CCR5 receptor is a kind of receptor which is found by macrophages and can be inhibited, which means it can be stopped to express within the macrophages, but C into CR4 co-receptor cannot be expressed, cannot be stopped, cannot be inhibited to express within the T helper cells, due to which it's often difficult to um, treat HIV as compared to other disease. Moreover, one more thing that I wanted to discuss over here, that here the virus HIV is basically having 9 genes within its double-stranded DNA. Double-stranded, sorry, it's not double-stranded DNA. Double-stranded DNA is formed within the cytoplasm. So before double-stranded DNA, its original viral genome is a single-stranded RNA. So here, as you can see, that as you can see that here we are having their DNA. So their DNA basically consists of around 4,960 nucleotides, 4,950 nucleotides, and apart from that, there are nine genes present. Out of these nine genes, three of the genes are responsible for their structural components, such as their envelope, lip, their as well as glycoprotein. Their lipid bilayer is host derived, whereas the glycoproteins are encoded by the genes. So, three of the genes are responsible for the structural support, for forming the enzymes as well as the proteinaceous capsid, matrix, and the glycoprotein spikes, while other structure while the six of the genes are responsible for replication and for function of the virus, which includes all of the steps within its life cycle, such as fusion, binding, attachment, replication, budding, and assembly. All of these are dependent on the function of these six genes. Moreover, um, there is some sort of replication initially of the viruses. I mean the HIV viruses when it enters the cell initially, but it shows no symptoms because uh, the symptoms are being combated by the antibodies present within our body, due to which the body is unable to treat it by itself. But once it reaches to the extreme level, chronic level, it is then not treatable much, and then it is supposed to be treated by medicines. And at that moment, it comes out as a detrimental disease. So this is how it comes out as a very dangerous disease in future after 8 to 10 years. And the treatment which is available for it is antiretroviral therapy, which we are going to discuss in next video. Hopefully you enjoyed this video. We'll meet again.